All right, we're back for part 2 of structural immunology. And here we're going to talk about antibodies first because they're what everyone has questions about, and they're also a therapeutic that's being made, a sort of drug, a protein drug that's made. We can make proteins. Proteins are all made of just 20 amino acids strung together in a certain order. We can do that. And so that means that we can make proteins that will actually bind to and act as antibodies in somebody's system. And there's actually a whole industry that's set up with making antibodies against cancer, against other things. In biochemistry, we're talking about one scientist who does this against cancer. But I want to say, before we get to that, what are the places where we can do it against COVID-19? Now, in part 1, we had this example of IL-33, which was the sort of magenta thing bound to the purple thing, the bluish-purple thing. IL-33 is the magenta thing, and if we have an antibody to it, the antibody can bind to it instead of the receptor, and we stop the signal that it sends. In the case of IL-33, it sends the poison ivy signal. And notice that that antibody is sort of a Y-shaped type thing, and I showed one part of it sticking to the, that's the way that it would work. It has a sticky end. And so an antibody is an immunoprotein. It's just a protein made of 20 amino acids plus some sugars on it. And it is designed and evolved by the immune system. Those are compatible terms, not mutually exclusive. An antibody's job is to bind an antigen that generates antibodies. An antigen can be anything. So a structure of an antibody has this sort of Y-shaped look to it. And when you look at it, so there's two different ways to talk about it. Sort of the schematic way on the left and the actual showing the protein backbone on the right. On the left, you're going to see that the antibody actually has two red chains with two boxes each. Each of those is an Ig domain, and it has a particular beta sandwich sort of structure that you can see. You can see how each of these sort of, they wiggle around, right? But they look pretty much the same. They have 8 arrows that are stacked on top of each other. Each of those is a box. And so each of these boxes here, you see that the red boxes have, the red chains have 2 boxes connected. The blue chains have 4 boxes connected. And they come together in this pattern to make this nice little Y shape. The important thing about the Y shape is that the bottom of it is just made of blue proteins, and those are constant. Those never change. That is the way that the body can know that it can bind to an antibody by grabbing onto that end of the molecule, the bottom of the molecule. On the other hand, the top of it, where you have the fingers of the antibody, and you notice that that says antigen binding site. It's made up of one red box and one blue box in each case. And the very tip of that is where the antibody contacts the antigen. All those things are amino acids, and they're made of atoms. And we learn about those in biochemistry. We even make those things in biochemistry. So there's various ways you can talk about an antibody. If you want to talk about it functionally, you talk about it on the left. You have the variable regions because the antibody has the ability to adapt to bind different things. And the places that adapt, the places that vary, are the variable regions, the binding regions. Then you have the places that are the handle. That's the constant region, and that's where the rest of the immune system grabs onto it. So you can talk about them functionally that way, or you can talk about them as the 4 different chains. The one with 2 boxes we call the light chain. The one with 4 boxes we call the heavy chain. And you'll notice it has a Y shape because of the way that you have the arrangement. 2 heavy chains come together, and they end up with 2 binding sites for each handle. 2 binding sites for each handle. It's a really important way for how antibodies work because you do get a twofer for them. You get 2 for the price of 1. And so that means if you have an antigen and you have a replicating antigen in your body, which means you have a bunch of it flowing through your system, each antibody can bind not just 1 antigen, but 2. If you have a repeating pattern on the surface of a bacterium, the antibody can bind 2 repeats of that pattern. It's buy one, get one binding, and it's one of the ways that antibodies work. I've actually seen the antibodies use this in the lab to have higher affinity. It's called the avidity effect. So you've probably heard of IgG, like gamma globulin. That is, that's a form of the antibody, the most common one, and it's called IgG. If you look at this, this has a table with the different characteristics of 5 different kinds of antibodies. The main characteristic I want you to notice is they all have this fundamental Y structure. Each Y has a certain size. They all have carbohydrates on them. Some have a little more, some have a little less in the second column. And they all have different concentrations in blood serum. IgG, high concentrations, it goes in the blood. Other ones like IgE, you have less of that concentration in the serum. It goes to other places in the body. And the other thing is that if you have something circling through the blood is a circulating dynamic system like we talked about in part 1. The antibody will break down after a time, and it will need to be filtered out and removed from the body. How long does an antibody last? Well, it turns out IgGs have a long half-life. Half of them are still around after 3 weeks, 21 days. So all of these are different characteristics, but you notice the biggest difference between these is that you have some that are just plain Ys, okay? And some that are 2 Ys stuck together, that's IgA. And then you have one that is 5 Ys stuck together, which has 10 binding sites per pentamer, okay? Each of these has different heavy chains, and the heavy chain, it determines the handle, so they have different handles. And they can be picked up by different parts of the immune system. These are different branches of molecules, just like we had different branches of the immune system. And so the different branches have different functions. For example, IgE is associated with skin and with immunity. And in fact, it's associated with overreaction of immunity. When you have your immune system overreacting to peanuts, it goes into your gut, it gets into your blood, it causes skin hives, because there are immune cells that are freaking out in your skin. They're sending out histamine, other chemicals that cause inflammation. If you look on those mast cells, what is the molecule that's making them freak out? It's IgE. And you see it looks just like the Y shape of the antibody. It's on the surface of mast cells in particular. And so it has a different function than IgG. It has a very similar structure. And IgE can have a binding surface. I can tell you the binding surfaces are right here and here for it. The cool thing is you can get into immunology and you can see all the different things antibodies can do. Once an antibody is developed, for example, you can have an antibody against a bacterial toxin on the left, and it can neutralize it by basically binding that toxin and keeping it from hitting the cells. You can have an antibody against bacteria that are outside cells, and they can actually grab onto the bacterium, and then a macrophage can grab onto the little blue handle and do what's called the process of opsonization and then phagocytosis to eat up, literally swallow up the offending bacterium. Then you also have another thing that's not cell mediated at all. Bacteria in plasma can actually activate this other system called complement, which is sort of like innate immunity, by the way, for those of you who are into immunology. Really cool, and it's entirely based on proteins. So you have cell-based systems that use the antibodies, you have protein-based systems. It's like a gigantic game of mousetrap, it looks like to me, if you've ever played that board game. All these complement proteins that come together, and they activate. Let me show you what the result of each of these is. Neutralization can keep the toxin from binding the receptor, and the yellow antibodies, they can then be ingested by a macrophage, or maybe they can just ingest the bacterium while it's still whole. Macrophages can do that too. That's the blue antibodies. And then the green antibodies show complement, which is also can be ingested by macrophages, but there are some parts of complement that are cell independent. They're actually protein systems. And I want you to see the little teal, hopefully you can see it above my head, the little teal proteins that are complement colored. They're teal colored, and they punch a hole in the bacterium. There are some like automatic pore forming proteins that they interact with the constant parts of antibodies, and they activate when you have an antibody bound, and they will activate to destroy a bacterium that way. They literally punch holes in the bacterium by forming a pore. This is one of my favorite artists, David Goodsell, and he shows immune recognition. You see a bacterium way over there on the left, and then you can even see the flagellum if you look at the top. And biochemistry is hopefully giving my students a feel, okay, there's the bacterial membrane. There's glycoproteins on it. This is a picture of an immune system in pink and purple attacking a bacterium. And by the way, blood proteins are in red. And so you can see that there are some fibrous blood proteins, but there's a lot of pink and purple. And if you look around, you can see the IGs. Try to look for a second to see if you can identify an IgG, which would be just a single Y shape. And then I can see an IgM or two. IgM is the one with 10 binding sites, 5 antibodies sort of linked together. And so if you look, you can see a couple of the IgMs sort of on the surface right there. You can see some pore-forming proteins in magenta that are breaking up that nice green bacterial membrane. And I just want to make the point from this. David Goodsell shows you how complicated and crowded and dynamic the whole system is. It's a messy, messy system. But you see here it's working. They're punching holes in the bacterium, they're binding to its surface, and they're alerting the immune system to this bacterium and hopefully kicking it out. Another thing you can learn about in immunology class, hopefully some of you are in it right now, is that antibodies are actually, how do you make so many antibodies that you have one on the shelf for pretty much anything that attacks your body? A normal functioning immune system will have a host of antibodies ready, and then it pulls that cell off the shelf and it makes clones of that cell. It's called clonal expansion. It sounds like science fiction, but the single cell with a single DNA sequence for making a single type of antibody gets amplified. The way that the body has so many antibodies on a shelf to choose from, so many naive B cells to choose from, is that it has a system for actually shuffling through genes like they're a bunch of cards. For example, right here, you have cells, you inherit like 8 gene segments that can make antibodies from your parents, let's say. Each of these cells takes these and it shuffles these together and draws a deck from them, draws like 4 cards from the deck. And the order of these matters, and there's even some mutation at the joints where they come together. But even just by shuffling these together, you can get 4 different colors, and you can make all sorts of different molecules by taking these 8 segments and shuffling them together. Once you have a whole bunch of different ones, you try them all out, and you take the one that works, and you make it grow. That is a description of how the immune system works. It also is a description about how evolution works. Evolution has gene shuffling, and natural selection selects one of those genes, and it has a survival advantage and expands. So this is one of the reasons why I basically am really fascinated with how evolution works, because I've seen the immune system evolves in essentially the same way that evolution itself has created species. When you look at antibodies, you can look at the binding region, the binding domain. Biochemists will see that this makes sense. It's about 100 residues long. It's a chain of 100 residues. And then you have most of those residues don't mutate that much. But then you have some hypermutable parts of it where you have them mutate more. And if you're the cell and you're making lots of antibodies, where do you want it to change more? Where do you want more mutations? Right at the tip of the Y, at the binding site. And in fact, if you look at these, you can take a whole bunch of antibody sequences and you can say, where do I have variability? You have a lot more variability in these 3 red segments over there. If you plot that out onto the domain structure of the antibody, those 3 red segments plot out exactly to these 3 loops. They're called CDR1, CDR2, and CDR3. It's because they are the regions that determine, that actually touch the antibody, the antigen. They're on the very end of the protein, these loops at the very end of each of the domains. They mutate like crazy. And the reason is, those are the parts that are actually touching the antigen. So you have all these different shapes of antibodies on hold, ready to be activated with all these mutations that make you have millions of different possibilities. And that's something that no pathogen can get away from for long. the antigen binding site is hypervariable. Okay. And so I do want to say, for my biochemists, presentation 4 is coming up. You can actually look at something about antibody evolution, because you can find things about antibody evolution that say something about evolution in general. Here is one exact example right here. This is a complex paper, but if you want to dive into it, my only request is that you type on the sign-up sheet that you are taking this paper, so no one else takes it either. But if you look at this paper, it's about how antibodies evolve, and they are guided by their intrinsic mutability. Okay, that you might get told that if something's evolving, it's random and purposeless. And there are some areas that are flowing and random, but there are other levels where you actually can gain a purpose built on top of layers that flow and are random. There's a case where it looks like this works with antibody evolution, and because it's so similar, by the way, biochemists, I said talk about evolution. Don't just assume I said talk about evolution of species. You can talk about evolution of antibodies as well. So the antibody has these domains. There's 3 loops per domain, and remember, 2 domains come together to make what's in the side view. It looks like this, and it has the 6 loops that are hypervariable. They can adopt almost any amino acid, and you basically see what works. We find out what the body has found that works. The end-on view, this is shown on the right, and you see how those 6 loops are arranged, and they make a surface. I like to think of this as, you know, the last thing the fish sees before the shark eats it. This is the last thing the antigen sees before the antibody binds it. It has these 6 loops that are kind of like the teeth that are coming at it. So the loops are making a binding surface that will stick to the antigen. And these loops are flexible. That means that the antibody can accommodate different sizes of antigen. If you have a small molecule, it can make a little divot. There's an example right here. I got this from a book. So it's hard to see where the divot is in this. It's the little orange thing in the middle of that. It can also make a groove, so it can bind a peptide. And this is a antibody that binds an HIV peptide. So clearly right here we have a viral connection right there. It can bind viral peptides by making a groove that fits the peptide like a 3D jigsaw puzzle. And then of course you can have a big surface that binds a big thing, and it can bind another protein surface. This is an example of an antibody binding a lysozyme protein, just sort of a test protein that we use to study how the immune system works. So the complementary interactions between the side chains, if you zoom in on the loops, each of those is an antibody, each of those is an amino acid, each of them has a side chain. And so you can zoom in on the side chains, and for example, here's one to a small molecule antigen called phosphorylcholine. Biochemists, you should be able to take that name apart and at least see, oh, it's got a choline positive charge, which is shown in pink, and it's got like this long the phosphoryl part. Oh, actually, I spoke too soon. The choline is shown in blue. The pink is the phosphoryl, and I can see that because I can see it looks like a phosphate group. I can also tell it's phosphoryl because look what it's binding to. It's binding to an arginine, which is positively charged. So you have plus-minus interaction. The antibody has set that up by having an arginine in the right mutation, heavy chain position 52 in this case. So all the structural stuff we've talked about as biochemists, we can apply it to antibodies, and we can use it to figure out how to fight off a virus. By the way, these loops can also move, and we have evidence for an induced fit binding mechanism, biochemists. For example, here is an antibody binding loop surface looking down on the surface with no antigen bound, and you're like, well, where's the binding surface there? This must be a flat antigen that is binding. No, when you actually look at the loops when you have antigen bound, and by the way, we're not showing the antigen, but you can see where it is. You can see that it's got this big gap right here. It might be binding a peptide or something about the size of a peptide. And that the antigen actually pushes the loops aside, and the flexibility is part of the antibody binding. So the loops are flexible. We can't tell, by the way, by looking at them if they're flexible or not. We have to do some kind of test because these are complex dynamic molecules. Oh yeah, and there's the antigen bound. Biochemists, this looks an awful lot like our case of the original Delano and Wells paper, by the way. This all ties together. We can break it down to the atoms, say what kind of bonds. For example, here's an antibody on the bottom binding to an antigen on the top. And this shows the hydrogen bonds. When you stretch them apart and look at them on a computer to see which atoms are touching the other ones, you see all these atoms are forming hydrogen bonds. They're like little ropes that are tying the antigen to the antibody. Well, maybe we can get an idea for a better hydrogen bond to make. Maybe we can design an antibody that forms more hydrogen bonds and makes more of a tight interaction with the antigen. We can help the immune system along. One thing to realize about antibodies, the body, we do have clonal expansion. Each B cell makes only one antibody, but there are a bunch of antibodies that are usually mutated. It's like what we're doing right now with vaccines. We're trying like 70 vaccines. We only really need one to work, but we have 70 candidates because we know some are going to fail, and so try a lot of things at once. The body does this with B cells. It makes one antibody, but it has a bunch of B cells, each of them making one antibody. And so the response of the body to an antigen, if you take all the antibodies out, you'll find a lot of different antibodies. They are polyclonal. That's really cool for the body, and that's the way the body does things. Scientists have to reduce the problem, and if we're working with one protein molecule, we need to work with one antibody at a time. Therefore, we take a polyclonal mixture of antibodies, and we figure out a way to pull out one antibody from that and get its amino acid sequence and be able to make more of it. That's called monoclonal. A monoclonal antibody is just a well-defined protein with a single amino acid sequence and a single structure that we can use. It's what we have to do, and if we're making a drug, polyclonal antibodies can be like the convalescent serum that you might have heard of. Monoclonal antibodies are the kind of things you can make in a vat. Okay, you have to have an organism making the polyclonals for you. We make monoclonals, and we can have whole factories to make monoclonal antibodies. I mentioned this before. If you have a drug that has AB at the end, if it has MAB at the end, by the way, that stands for monoclonal antibody. And you can see we have a whole bunch of different ones. I think I'm covering up a little bit of this, sorry about that, but you have a whole bunch of rituximab is an antibody against an antigen that's in non-Hodgkin's lymphoma. It treats that. You have antibodies that treat rheumatoid arthritis, antibodies that treat asthma. They basically go against different parts, and look at the one on top. It's called Herceptin. That's its commercial name. Its generic name is trastuzumab, and that goes against breast cancer. We actually will talk about that because James Wells works for the company that made that molecule. And actually, if these always are proteins, so it's hard to take them orally. You have to inject them straight into the blood. But they are very effective. Herceptin has saved a lot of lives and killed a lot of cancer cells, you know. There's actually a whole nomenclature for which system it targets and which source it comes from. That's why these antibody names are so hard to say. As you become a doctor, you can get more into that. For right now, let's just say if it ends in MAB, it's a monoclonal. And so you might already be thinking, oh, I sure hope people are doing this against COVID. And they are. We can form antibodies against the red viral spike proteins. Here's a cartoon of COVID. And I'm going to attribute the author, by the way, in a later part. We can make antibodies against that, and they can interfere with the virus, or maybe alert the immune system to the virus. And here's a bunch of papers. I'm just going to show you those papers. Biochemists can see a little more detail in these. Here's a human monoclonal that blocks SARS-CoV-2 infection. What they do is they add the antibody, and the blue lines are where they add the antibody. As they add more of the MAB, you see that on the x-axis, that they actually end up blocking SARS-CoV on the left. That's the one that caused the SARS epidemic in 2002-2003. It's the closest virus to SARS-CoV-2. The good news is, because the viruses are so close, the antibodies work against the same. Some of the antibodies work against the same proteins, work against both viruses. Here's a monoclonal. They had 51 monoclonals that they tried, hybridomas that they tried technically. And they found out that 4 of those displayed, 4 of those ones that went against the 2002 SARS goes against the 2020 SARS. And so the evidence is right here, that blue line going down. By the way, the interesting thing is they found out this monoclonal, they looked at where it binds. A lot of monoclonals block the receptor binding site. That's what you expect. That's the simplest way to stop a virus. But not all of them do. And this particular antibody actually does not block the receptor binding site directly. They conclude that it neutralizes, it stops the virus through some other mechanism. It knows now, and we've got to figure it out. But it's figured out something. And so the more we study it, its structure, we can figure out what the atoms do, and we can replicate those atoms. We can grow this antibody in vats. I'm sure there are people that are doing that right now. And we have to test it to make sure it's safe first. So here's another one. This is from a review. You see the title at the bottom. We have an antibody that binds the spike protein. Here's the spike protein sequence, by the way, biochemists. You can see all the amino acids that we learned. And above the box you have blue is similar residues, red is different residues, and sort of beige is exactly the same between SARS and SARS-2. So you can see that these are indeed very similar. You do have some distinct differences, but we have some antibodies that don't care about those differences. Here's an example of an antibody bound to the RBD, or the receptor binding domain. That's what's shown with the spheres, the little balls that are up, that the red antibody is binding to. So here we can go in, and they've plotted out with the purple spheres in the spike protein. You see that there's a couple of purple spheres in there. Those are the non-synonymous mutations. Those are the differences between SARS-CoV-1 and SARS-CoV-2. There's not many of them. So it makes sense that some of the SARS-CoV antibodies that we've kept in the freezer can actually come out of the freezer and are helping us against SARS-CoV-2. These are all proteins, these are all atoms, we know where they are, and we can make them. Here's a little more detail about that. I just want to say that efforts are underway, including phage library display. I put that in for my biochemists. Then you do traditional things where you immunize a mouse, and then you make a hybridoma out of that. And then you can actually take human B cells that have fought it off, and you can clone those. And so the thing I want to say is there's a little bit over there. We can take the spike protein, and we can make that in a lab. And then we can use that to generate antibodies. And so there's some text over here about how that works. And they give some thought. Okay, SARS-CoV-2 has mutated in these purple places relative to SARS-CoV-1. How do we know where can it mutate again? Where is it already mutating among the different strains? There's only 4 known alterations in the protein at the amino acid level at the time they wrote this. There they are right there. We can plot them out on the spike protein, and we can see. Notice that there's only 4 residues out of that whole big protein. It seems that the virus is trying to mutate, but the chances of one of these mutations significantly affecting its function are hopefully pretty low still. It just hasn't had time to mutate that much. So that's one of the good news, but we can also look at these and we can compare them to similar mutations in MERS and the original SARS. And that's what they're doing here. MERS and SARS, the research that we put into those is really paying off right now, because we can do things like take research against SARS-CoV-1 and apply it to SARS-CoV-2. Here's something that my friend just put up last night on Instagram, and I was like, I can't believe that you're tweeting about what I'm going to talk about tomorrow. A 4-year-old llama named Winter could hold the key to a cure. Okay, so this is my friend Holly, and she posted this, and yeah, she was kind of surprised. And I was probably the only person who was not surprised by this, because I've heard a long time about how llamas have extra special antibodies. They're extra special because they're simple. And they're also, I believe, they're robust, or it's easier to make them robust. But the main thing is, they are simpler than the Y-shaped human antibody and mouse antibody shapes that I showed you. And so people are immunizing those llamas and making llama antibodies that are easier for us to make in a factory because they are simpler molecules. And they also can be made more stable. So here's the paper that this was talking about. This raised antibodies from camelids, which is llamas. And the antibody is in blue. It binds SARS-CoV-1, the receptor binding domain. But you see it binds a part of the SARS-CoV-1 that doesn't have much difference between SARS-CoV-2. And so they started off with this knowledge. By the way, the red at the top is the receptor binding interface. The receptor is called ACE2. And you see here they have another case where they have side-on binding, where they have it binding to the side. It's not directly blocking the receptor. Actually, that's good sometimes. So we're trying to figure out how that works. We can zoom in on the interface. You can see the blue antibody loops, and they're forming hydrogen bonds and other interactions with the pink SARS-CoV-1 receptor binding domain. And then what they did is they took this out and they said SARS-CoV-2 looks pretty much the same as SARS-CoV-1 for the residues that this antibody touches. Let's see if it binds. And then they showed this. This is what we do in my lab. In physical chemistry class, we actually are measuring exactly like this, things to show that it does bind. They took the SARS-CoV-2 receptor binding domain, and they detect the binding. The red lines going up means they detect binding with a tight KD. Biochemists, you will notice this is a 40 nanomolar interaction. That is tight binding. We can also say the kinetics of the interaction. If you want to know more about that, take my physical chemistry class. And that is what we can do for SARS-CoV-2. Now I will transition to how we can use them against antibodies, and I'll tie this in for the biochemists.